Hello everyone, welcome back to class. In previous lessons, we studied the internal environment for cell survival, methods of homeostatic regulation and control systems. These are the environments in which cells live. Today we begin to analyze the structure and function of cells to see which universal basic functions make up our cells. If we view the body as a planet where cells live, each cell resembles an independent individual. Every person has skin covering and protecting them. So what covers the outside of a cell? Is it similar to human skin? Let's explore the cell's exterior together. The cell membrane, also called the plasmalemma, is a membrane that separates the cytoplasm from the surrounding environment, about 7 to 8 nanometers thick. It is mainly composed of lipids and proteins, with a small amount of carbohydrates. How do these three substances form the membrane? The widely accepted explanation is the 1972 fluid mosaic model proposed by Singer and Nicholson. This model states that the membrane scaffold is formed by a fluid lipid bilayer, in which various membrane proteins are embedded. Carbohydrate molecules bind to lipids or proteins and attach to the outer surface of the membrane. Phospholipids are the predominant lipids in the membrane skeleton, accounting for about 70% of total lipids. Cholesterol is next, under 30%. Glycolipids are present in the smallest amount. Lipid molecules are amphipathic, with a hydrophilic end and a hydrophobic end. The phosphate head of phospholipids and the hydroxyl group of cholesterol form the hydrophilic end. The fatty acid chains are hydrophobic. Both inside and outside the cell are aqueous environments. So amphipathic lipids arrange in a bilayer, with hydrophilic heads facing extracellular fluid or cytosol, and hydrophobic tails facing each other, forming an internal hydrophobic region. This structure is crucial for restricting free movement of substances. Next are membrane proteins, which carry out most functions of the cell membrane based on how they are present in the membrane. Membrane proteins are classified as peripheral or integral proteins. Peripheral proteins make up 20% to 30% of membrane proteins and are easily detached. Integral proteins constitute 70% to 80% and are tightly associated, removable only by disrupting lipid structures. The protein-to-lipid ratio varies widely among cell types. Functionally active cells generally have higher membrane protein content, whereas simpler cells have less. For example, in small intestinal mucosal epithelial cell membranes, the weight ratio of protein to lipid can be as high as 4.6 to 1. In Schwann cell membranes that form myelin, the ratio is only 0.25 to 1. Finally, carbohydrates are present in the smallest amount. Although low in quantity, they play indispensable roles. Carbohydrates in the membrane are mainly oligosaccharide and polysaccharide chains, which bind to proteins or lipids on the extracellular side, forming glycoproteins or glycolipids, known as the cell's antennae. They serve as molecular markers, acting as receptors or antigens. For example, the ABO blood group antigens on red blood cell membranes are determined by different carbohydrate chains. Thus, carbohydrate sequences act as a molecular language. The cell membrane is the skin of the cell. Besides maintaining the cell's internal microenvironment, it also participates in material exchange and signal transduction. It plays an important role in normal cellular physiology.